This is visible now?
Yes, ma'am.
Yes, ma'am.
Okay. Sorry for the inconvenience. Yeah. So we were here. So we have actually checked again the cytotoxicity of polymers. You can see that in case of epsilon polylysin and linear polyethylene amine, the cells were completely safe as compared like control untreated. The cells are very healthy, but you can see the PLL. In this case, the cells, you see red color and the cells are circular. That means the cells are completely dead. So this is how basically we demarcate. Here the proper proteins of the cells are visible, the nucleus in blue color is visible, and they're properly proliferated. So the cells are healthy. But in this case, this material is PLL is basically cytotoxic. That's why the cells shows circular morphology. That's why what I want to show here. So then, once we came to know, okay, EPL, epsilon polylysin, is absolutely a safe polymer. We can use it for as antimicrobial cationic polymer. We actually studied its antimicrobial properties in details. You can see here uh, different kind of pathogens we have investigated, that for which kind of bacteria it is effective. But it is surprising that it has very low MIC values for most of the bacterial strains, means it can kill majority of the bacterial strains, whether they are gram positive, gram negative, or resistant strains. Also it is very effective, and even the fusarium, which is fungus. So we came to understand that the EPL, epsilon polylysin, is a cationic polymer which has a broad spectrum antimicrobial properties. And one interesting properties I want to share, that in Japan and Korea, this epsilon polylysin, it is used as a food, food additive, or you can say food preservative. Why? Because it has antimicrobial properties, so even people are eating this polymer. So we thought, okay, it is, that means it is quite safe. Dermal cells we have already tested, and it is used as a food preservative. That means it is completely safe polymer to use for a medical material. So we actually same way we have designed the epsilon polylysin based dressings on the bandage gauge. We have designed the dressings. We have checked its antimicrobial, the disdiffusion zone of inhibition. I already explained you. You can see here clear zone for all the strains we got for this particular polymers. And you can see this is, I think for students, it must be very interesting to see the human cells, how they look. There are two type of cells you can see here. This A picture, it shows the keratinocyte, skin keratinocyte cells. In the B picture, you can see the human dermal fibroblast cells. You can see how the cells at the 4-day and the 7-day, how the density increases. In the SCM pictures also, you can see how the cells look in the SCM micrography. So the cells were growing very happily on these dressings, that we understand. Even in the in vivo studies, we find that the progress of the wound closure is much better in epsilon polylysin as compared to the commercially available dressings. This is the histology study. This shows that how, when there's a burn, the upper layer is completely sacrificed, and when we use the EPL dressings, how the skin cells they grow and maintain its morphology. In the last SCM picture, you can see these circular cells attached. So basically, this is the SCM picture of the dressing after removal from the wound. So you can see once it is removed from the wound, the microbes, they are completely attached on the dressings. So it works in that way also. It kills the cells, and the dead cells, they actually attach to the dressing, so even it help in the cleaning the, or debris, whatever is there, it is help in cleaning the debris also. So this is the second part of the work. In the third part of this same work, what we did with gelatin and polydopamine, we have used antimicrobial metal ions. So before starting with the Mendel lines, we basically went to the literature and we actually identified some metal ions which has antimicrobial properties and also safe for human cells. So we identified four metal ions: silver, magnesium, calcium, zinc. Why we have taken? Because these metal ions are normally very important for our biological system. Usually we take zinc supplement, calcium supplement, magnesium is also there as a part of our hemoglobin. So we took those metal ions which we know that they are safe for our body, and silver anyways people are already using it, so we have used it. So we have integrated these metal ions in the dressings, and you can see the antimicrobial properties. Pure gelatin, no zone. Silver, beautiful zone. Calcium is effective for gram positive bacteria, but for gram negative like Pseudomonas, it is not working. For magnesium, it shows zone of inhibition, good zone for positive, but for gram negative, it is inhibiting, but not so much. And zinc is also good for all, but better for gram positive. And you see the cells, how happily these cells are growing. So even with these metal ions, we got the antimicrobial properties, and even they are safe for the human cells. That's what this slide shows. But you see here one important information I want to give. If we don't use polydopamine as a cross linker, these dressings are not very safe for the human cells. You can see the in the upper panel, we have not cross-linked it with polydopamine. We have only gelatin and silver, gelatin with magnesium. So you can see the cells, they are in the circular morphology. Even in the other cases also, the cells are not very healthy. But when we did polydopamine cross-linking in the lower panel, you see the cells are happily growing, and they're completely safe. So this shows the importance of polydopamine in slowly releasing any antimicrobial component. So we actually check the SCM picture to understand the reason. So when we don't have polydopamine, the metal ions are actually, on the surface. But when we have polydopamine, it is integrated inside the surface. So that is why there is a slow release in the polydopamine cross-linked nanofibers. And this shows the long-term antimicrobial activity of, this our magnesium-based dressings. Till 40 days, it has antimicrobial properties. So this is the conclusion, that we actually discovered safe and efficient cross-linker for hydrogel electrospun polymers. Antibiotics in combination with polydopamine crosslinker can be used to design durable antimicrobial wound dressing with long-term antimicrobial properties. The method did not interfere with the antimicrobial properties of the drugs. This strategy can be extended to metal ions. It can be extended to the other antimicrobial compounds also. Polyphenols with